I'm Sharon Todd. I'm the CEO of SCI, which is the Society of Chemical Industry, and it's my pleasure to welcome you all this evening and introduce our speaker this evening as well. Before we start, I have to give you the safety briefing, which is that we are not expecting a fire alarm tonight. So if the fire alarm goes off, you do need to do something. And what you need to do is exit back up the stairs where you came in, or exit through this fire door here. And there's a corner on Belgrave Square, which is where you need to meet, and we will look after you. The team will be around, and please exit as soon as possible. So that's if there's a fire alarm tonight. And also we ask people to switch phones to silent, so that we're not distracting the speaker. Please do not take photographs during the lecture. It can be quite distracting, and we are streaming as well, so just to be aware of that to other locations. We do have a Wi-Fi, yes. Don't know it off the top of my head, I'm afraid. There you go. Somebody like to pass that back to the gentleman who needs the Wi-Fi. Fabulous. Feel free to send tweets out on Wi-Fi. Yeah. Right, so a few points about SCI. So SCI is officially called the Society of Chemical Industry. We were established as a society in 1881, and we were established for a very interesting mix of scientists, inventors and entrepreneurs. I'll come back to that because that mix was actually quite an important mix when the society was put together. We are a very unusual society. We're what's called multi-science and multidisciplinary, so we cover a wide variety of science areas, but with chemistry at the core, and we cover a wide range of disciplines as well. And the reason why we were set up was really to help the translation of science into industry for the benefit of society, because it's only when science goes to industry that it actually becomes available to society. So that's very much around fostering innovation and supporting the application and the commercialisation of science via new products and processes. So in our history, still today, we have what I would call prominent scientists, so Nobel Prize winners. So who knows that this year is the year of the periodic table? I've got a lot of chemists in the room, that's great to hear. Sorry, I'm struggling a bit with the technology, it seems to be flipping around. So William Ramsay, who you probably know, discovered a whole line on the periodic table, the noble gases. Lord Todd, sadly not my relation, and more recently Harry Kroto, who I'm sure some of you would know, long-time member of SCI. So they really pushed the boundaries of the science forward in the purest sense. And then the inventors, who are often the sort of forgotten people, often the industrial scientists, create things out of the science. So people like Leo Baekeland created Bakelite. I think half the room might remember Bakelite, but the other half definitely won't remember Bakelite. Those old telephones from my era, that was really the start of the synthetic plastic and materials sort of revolution. And then people like Richard Seligman as well. So there's the scientists, there's the people that do things with the science, and then there's a whole bunch of entrepreneurs, the people from Silicon Valley, who are actually then creating businesses and putting these products out to society. Probably too busy to read, but just to note really that this kind of collaboration and collaborative approach is very important. So in our founding fathers, we had members from Pilkington's family, the glassmakers, Henry Tate from Tate and Lyle, the sugar makers, all the founders of companies like ICI, Procter & Gamble, Unilever, and they all came together with the scientists and the inventors to really help promote science-based innovation into industry for the benefit of society. So today, SCI carries out a number of things. We support open innovation networks, innovation skilling through our skills and education programme for early careers and mid-careers. We run publications, scientific publications, but also publications that are more geared to the general public. We have a whole set of activities around recognition and making sure people understand the value of innovation, and innovation policy and strategy in particular around things like the industrial strategy in the UK today. So lots and lots of things that we do, and if you're interested, please contact us and get engaged. So that's a little bit about SCI, but now it's my pleasure really to do what I'm here to do, which is to introduce our speaker this evening. I'm delighted to welcome Professor Paul Workman, who I know is going to give us a fabulous talk tonight, and we're very pleased to have secured him as a speaker. He's a difficult man to get hold of and book in, but I think we managed it, and I'm sure you're going to have a real treat tonight. So just a couple of things, because I think may be reflective of the piece I've just talked about there, struggling again with the technology. He is the Chief Executive and President of the Institute of Cancer Research, and I know you're going to be very interested in what he has to say tonight. This is a topic that really affects all our lives and is an important one, and I think he's going to take you through some of the groundbreaking research that they do and also the products that they've helped create. He has a biochemistry degree and also a PhD in cancer pharmacology, so he comes from a kind of background that we're familiar with. And he very interestingly has both an academic and an industrial background, both with large companies such as AstraZeneca as well as startups and entrepreneurial companies as well. He is a serial entrepreneur, and he's been through that loop very many times, and he's interested in multidisciplinary approaches. So all of those things, I think, make him very well suited to talk to us this evening, and also make him very well suited to become part of the SCI network, because this is very reflective of the work that SCI does. So I know you're going to be very interested to hear him talk, and it's my absolute pleasure. I would like you to join me in welcoming Professor Paul Workman tonight. Thank you.
Thank you very much. Thank you for the kind introductions. Thank you all for coming out. I nearly didn't make it this evening because I was involved in a fundraising event for my institute in Cumbria, from whence I come. And we jump up and down the mountains once a year to raise money. At the end of the event, there's large amounts of beer drunk, and an auction is held. And at this auction, I regularly purchase or bid for, and successfully bid for, tickets to watch a certain football team that I support, which is called Manchester United. I realise I probably lost a few friends. Manchester United were due to play Crystal Palace last night, and the game was mixed, but was changed to tonight. So rather than stand you up, my son and his friend are attending. No doubt Crystal Palace beating Manchester United at Selhurst Park. So anyway, it's a pleasure to be here, and it's a great society, and happy to speak. So recognise this as a mixed audience, lay, probably some chemists, maybe some biologists of various types. So I'll try and get the messages over, first of all, about what cancer is. What is this disease called cancer? Why is it a problem? What are the challenges that we face? It's a talk that's about hope as well, because it's about how we understand cancer and treat it better, but it is one about, you know, the language about the war on cancer and doing it overnight and having a single miracle cure is really something that we shouldn't expect. So it's being pragmatic and realistic, but ambitious, and there is a strong message that we can really do things, and we are doing things for cancer patients. I'll also really illustrate this talk with work that we've largely done at our own institute, but it will represent the work of a whole community internationally. So I want to start by I won't be doing this very often, rest assured, but I just want a small contribution from the audience at this point. I'd just like you to raise your hand, if you would, if either you or a close family member or a friend has been touched by cancer. If you look around the room, I think you'll see that that's the vast majority of people either been touched by cancer now or almost certainly will be at some point in the future. I always find, I do that quite often. I find it a very salutary, every time I see it, I find it a very salutary thing to see. So, yes, I have to go down, not up, that's the, yeah. So at the Institute of Cancer Research, our mission is to make the discoveries that defeat cancer. And in this mission, we really state two things, which is that we have to discover things about cancer itself, and then we have to, that's a cancer cell, by the way, in the middle. You'll see some quite interesting images, some rather beautiful, but also terrifying images as we go through. These images actually come from a photography and imaging competition that we have for scientists across the Institute. And each year we have that judged externally and also by the public vote. And I'll be showing you some of the images that illustrate cancer from that event that we've done. It has a very strong outreach because the images are very powerful and show meaning as well as being very interesting to look at. So our mission then is to understand cancer, but then to take it through, in contrast to some basic research institutes, we actually do drug discovery, we do targeted radiotherapy research, and with our partner in the Royal Marsden, we take our therapies through into first-in-human trials and then through into the NHS. So it's a bench-to-bedside approach that we do. Just a little bit about me. You heard just a little bit of the background, but just a little bit more detail. So I have the privilege to lead the ICR, which is a college of the University of London, a cancer research institute in its own right. We teach PhD students, as you'll see in a moment. And I've been leading this organization since 2014. But when I left AstraZeneca in 1997, when I looked like this, I became director of the then Cancer Therapeutics Unit, or still is the Cancer Therapeutics Unit. I took over what was a very active and successful cytotoxic drug development group, a drug discovery group, and morphed it into a molecularly based biology grouping that then linked in with modern technical approaches, robotics and high throughput approaches, structure-based drug design and so on, and built it into a successful group, which you'll see some of the things that are coming through from that. But before that, I did have a very enjoyable and successful period at AstraZeneca, where I learned a lot about scale and industry and project management and so on. That was very useful. I found it throughout my career to be very interesting and useful to come in and out of the three sectors really with biotech as well. So you can see before that I was at Glasgow and for a very long time at Cambridge in the oncology unit at Cambridge, and briefly at Stanford in California on sabbatical. I founded two biotech companies, Chroma Therapeutics and Pyramid. Pyramid was acquired by Roche. And I've advised a number of other organizations, as you can tell. So my personal passion is understanding cancer biology and turning that into drugs, understanding how those drugs work. And I don't expect you to read all of this, but I am required in these days, and it's quite right to do so, that to share with you the various companies that I have interacted with. It's not all of these now, but this is the full listing of that. But I'll show very clearly where we are successfully linking with companies, and I think that's a strength. So let's just talk a little bit about cancer. What's the scale of the challenge? Well, by 2020, 1 in 2 people will get cancer. So I think, fairly soon it won't be when I ask that question about how many people are touched by cancer in the room. The 50% will be those who actually have cancer or have survived cancer. So it's very common. 8.2 million deaths per annum worldwide. 166,000 deaths per annum in the UK. That constitutes a quarter of all UK deaths. And if you add all the cancers together, cancer as a group of diseases, of which there are 200, by the way, at least, now being subdivided by molecular genetics, it is the most common cause of death across all cancers in the UK, more than cardiovascular disease, in fact. New cancer is diagnosed every 2 minutes. I find that a very useful statistic, a powerful one. Very, very common, very frequent. But over 50% of patients now survive 10 years or more. That's a doubling from, if you look back over the last 4 decades, which has been a real period of increase. So more than 50% of cancers, 10-year survival is generally equated with cure. So that's a fantastic, I think, achievement from cancer research. It's been based on cancer research and cancer treatment. But the survival for many cancers, within that figure lies a host of mixed statistics, actually. So I can illustrate that here with data from Cancer Research UK, who fund our research and is a great organization. So this shows in the pink the female cancers. It's a bit busy, but I'll just explain to you that on the left you've got the female, on the right you've got the male cancers. The types of cancer go down vertically, and the 3 lines across each cancer type are just whether it's the 10, 5 or 1 year, but they're all fairly close. You can just look across and see that these days, if you get a malignant melanoma, which is the skin cancer you get from sunburn, the cure rate at 10 years is now, where it used to be dismal because of the new BRAF inhibitors and the immune drugs, it's now out at 80 to 90%. Surgery, of course, is incredibly effective if you can treat it, melanoma early before it spreads. Breast cancer in women and prostate cancer in men, the survival rates are very high, as you can see. Bowel cancer is in about the middle for many. If you go down, you'll see brain cancer is dismal. Pancreatic cancer is very poor. Lung cancer is poor. Esophageal, that's windpipe cancers. So there you can see is that some cancers do very well, other cancers do less well. I think some of the cancers that are at the bottom have been not so well studied, and the reason why perhaps it's argued breast and prostate and other cancers like that do better is because strong patient advocate groups, strong research into those areas, and that's been where the light has been shining. We need to shine the light into the other areas, and we and Cancer Research UK and many others are making sure that we try to do that. But you'll see that the drugs that are coming through in some of these poor outcome cancers, is in a report that we published, which I'll show you at the end, from ICR, showed that there just isn't enough work going on. It's not that cancer drugs are being developed for these poor outcome cancers and they're not working. It's simply that they're not being developed and taken through. So there's a policy message in this as well. And what you'll hear is that the notion that you have a particular cancer drug and it will work across all cancers is long gone. Cancers work and are now designed to work in particular types of cancer, and not only as labeled by where the tumor arises, but actually what the molecular makeup of that cancer is, and that's how the drug is designed in order to overcome a particular molecular dyswiring or functional aberration caused by DNA damage and mutation of the DNA. So let's just look quickly at how cancer develops. So if you just imagine this is the lining of your gut, so you've got the gut is at the top, there's the gap, the lumen of the gut, and you've got the pink cells are just the normal cells all lined up growing nicely, properly, and then you've got some early cancer cells growing there, and you can see it's more disordered and they are a different shape and they are starting to bulge out of the local environment. So there's uncontrolled, abnormal appearance and uncontrolled growth. They divide when they shouldn't and don't stop dividing when they should. They grow out of control into, you can see them spreading locally, that's called invasion. They're spreading into the adjacent organs, and so you've got uncontrolled growth that then invades into the local, so that's local spread. And then the next stage is that through the cancer cells being shed into the blood and into the lymphatic system, there's cancer cells spread around the body, and that's where you can see, so that's the spread of cancer, sometimes called by the experts metastasis. So that's a very simple view of what cancer is. It's a disease that's characterized by uncontrolled cell division, cells dividing when they shouldn't, and cells then spreading around the body. And oncology is the term that's the study of cancer. So when you see an oncologist or an oncology researcher, that's the name that's given to the study and treatment of cancer. So here's your first picture of a cancer cell. Actually, it's a cancer cell that's in the process of dividing into two. It's a lung cancer. Actually, this came from Anne Weston's work at the Crick Institute, who we work very closely with. So it's a lung cancer cell that's dividing into two when it shouldn't. Obviously, it's doing this in isolation in a test tube, but this is exactly what goes on. So it illustrates the first point, which is cancer cells grow and divide when they shouldn't. And then here's what cancer cells do is they sense the environment in a different way to the normal cell. So the normal cell will get a signal. It puts out chemical signals called from adhesion molecules on the surface of the membrane, touching other cells. When it touches another normal cell, if you're a normal cell, you stop dividing. If you're a cancer cell, you touch an adjacent normal cell, you ignore it, and you continue to divide and to spread. So here's a beautiful picture, terrifying as well, isn't it, of a triple negative breast cancer, which is one of the most poor outcome and the cancers. And the colors are the different adhesion molecules located around the so-called focal adhesions. Now let's look at what this does. So the cancer cell, so now you're seeing obviously a time lapse of a cancer cell sensing its environment. I could have shown you a picture next to other cells, but it's much easier to see the cancer cell sensing, putting out signals, sensing the local environment and whether it's got time to spread to, space and the ability to spread into adjacent tissue. And this is a amazing picture. I think it was the winner from the expert panel chose this as the top picture image from our annual competition. And this cell, sorry, just go back. This cell here, the pink one, the DNA is in the middle, and what you can see is the DNA is condensed. It's in the process of dividing. So it's going to do what the previous cell did, which is divide into two. But at the same time, these other cells are blood vessels. So this is not only dividing, which is a scary thing, because you don't want the cancer cell to divide, but it's also traveling through the blood vessel while it's dividing. So it's a double sort of terrifying image in that way, but a fantastic one, I think, to capture. This was done by Dr. Lam in our institute. So you've got uncontrolled growth, invasion, and then eventual spread around the body. Most patients, if they have their primary tumor taken out, which is the local one, will survive. It's the ones that invade locally and then spread throughout the body that are the ones that usually kill the patient. So there are three approaches to tackle cancer. I will be talking mainly about treatment, actually, because it's what I do, but my institute does all of these. You can either prevent cancer, which is ideally what you'd like to do, of course. You can detect it early, diagnose it, and then you need an improved treatment to overcome the cancer that you detect. So that's broadly where cancer research is largely focused. I'm mainly a therapist type of scientist, but I have colleagues working in the institute and elsewhere that work on these other aspects as well. They're all important. So let me introduce ICR. The Institute of Cancer Research on the Fulham Road in Chelsea and in Sutton is an independent cancer research institute. We've got about 1,100 staff and students, 80 faculty. We're a college of the University of London. We teach PhD, MD and MSc students. The government doesn't, every few years or so, decides to do a quality assessment of all its universities, and we've had the good fortune to come top. Obviously in the small areas in which we work, or the important but finite areas in which we work. So our research quality is high, as judged by that type of peer review. And with our Royal Marsden colleagues, so the basic research, I think I should use that, shouldn't I? Yeah, so basic research in biology and genetics of cancer, understanding what cancer is and how it's caused, then coming up with new treatments and markers and tests for detecting it, translating through to new therapies, radiation as well, taking them through across the road into the hospital, doing early stage trials in the first in human trials, and then feeding back research back into the basic biology lab. So with the Royal Marsden, we're one of the top what's called comprehensive cancer centres in the world, which means by comprehensive cancer centre, that is, you go all the way from basic research, applied research, coming up with new treatments, early clinical trials, and then care for patients, in our case in the NHS. And I mentioned that we're located both in London, Chelsea and in Sutton. And I was just explaining earlier to colleagues at SCI that for those of you who know about the Royal Society of Chemistry's blue plaque system, we're very privileged to be given 2 blue plaques just in the last few months. I should have brought a picture. But for our drug discovery work, we have a blue plaque which will go outside our Chelsea site in Fulham Road, and a blue plaque that will go outside our Sutton site. So that's very nice to have from Royal Society of Chemistry. So what have we done? As I say, we do well that's all very well to be ranked for the quality of our research by the papers that we publish, but we were actually the first to propose the link. So Ken Away, who was one of the first directors of my predecessors, was the first to propose the link between smoking and lung cancer, and then Richard Dole and Julian Peto and colleagues did the prospective studies that showed that it was indeed the case. And we went on to show, having identified carcinogens present in coal tar and so on, we went on to show the first evidence that the cause of cancer is damage to the DNA. So cancer is a disease of genes. It's a disease of mutations, and it's the mutations that cause cancer. And that's led to the diagnosis of cancer being now very genetic, and the treatments that we now develop are very based on whatever genes are particularly wrong and responsible for any one particular cancer. But it's all buried in the history, or based in the history, I should say, of that work. Since then, having discovered that cancer DNA mutations are the cause of cancer, we then went on to find many of the genes that suffer from these mutations and then have abnormal growth, because these genes are regulatory genes, and if they go wrong, the cancer cell grows abnormally and doesn't respond properly. And so these are some of the genes that we've discovered: NRAS, BRAF, and the BRCA2 gene. That's one of the Angelina Jolie genes, so-called, for familial breast and ovarian cancer. We identified that. Probably less well known than our drugs, actually, is that we've made major contributions to, I'm so sorry, I've pulled that back, major contributions to the development of high-precision radiotherapy. I think radiotherapy gets a bad rap. People always want to hear about our latest drugs, but as I'll show you later, the incredible technical developments in radiotherapy mean that more than 50% of patients treated with radiotherapy are cured of their local cancers. Obviously, it doesn't treat cancer that's spread around the body. And we developed techniques that are called MRT and IGRT, so intensity modulated radiotherapy and image guided radiotherapy, that make the radiation beam much more focused on the tumor and sparing the normal tissue. I'll tell you more about that later. But I think probably most well known perhaps of what we do is our discovery of cancer drugs. So melphalan, chlorambucil, and carboplatin in the early days, abiraterone, and this drug, which I'll tell you about, doesn't have a name yet. I'll tell you more about our drugs and how we work very closely with industry and biotech to take those through to patients. We also have made a major contribution over the years to immunotherapy, and that's the new hope for cancer treatment, activation of the immune response to cancers. And Jacques Miller, when he was a PhD student at our institute, was the first to discover the function of the thymus. It was thought at the time that the thymus was a redundant gland that had no role, like the appendix. Of course, the role of the thymus in the immune response is now incredibly well known and forms the basis for modern immunology and the new checkpoint drugs that I'll tell you about later on. So that's a bit about our history, and this is about our working with the Royal Marsden. So it's a unique partnership, bench to bedside. Here's a scientist working in the, sorry, I'm doing that again. Here's a scientist working at the bench, and just a few yards away, here's a patient being treated, perhaps with one of our new drugs in the Royal Marsden. It's that coming together of a powerful cancer institute and a powerful cancer hospital working in tandem that makes, I think, us quite effective. So this is our mission, or our vision actually, in research strategy. If you want to read more about our research strategy, it's on our website. There's a PDF you can click on. You only need to put ICR and research strategy into Google and you'll get the document. You don't need the whole thing. So our vision is that we'll overcome the challenges posed by here I'm now going to introduce a concept that's critically important to understanding cancer and the challenges of treating it, which is cancer's complexity. On the one hand, just like normal cell division, there are some basic rules in biology that apply, across plants and normal cells and indeed cancer cells, how they divide, how they move, and so on. But cancer's complex in the way that it dysregulates those simple rules and many complex ways that it has to get around the blocks that normally stop cells from dividing wrongly or spreading incorrectly or traveling around the body. So that complexity, and moreover, as I'll show you, and remarkably, if you haven't come across this, I still find it remarkable and I work on it all the time, cancer can adapt in response to treatment, and it can evolve through actual Darwinian type evolution, albeit within the individual patient, not across species. But the ability to evolve and become resistant to the treatment is, in my view, the major challenge that we face. And so our mission says that we'll tackle that by being excellent in the science and the clinical approaches of cancer, by being innovative, which often means working with small startup companies, creating companies, getting things out into bigger companies sometimes, and then partnering with patient communities, with industry, in an ecosystem that I think is really essential if we're going to crack this tremendous challenge of cancer that we all face. So this is a slightly complicated slide, but the biologists might recognize that if you were to take if you imagine that it's a single cell that has DNA damage, maybe from sunlight, and creates a melanoma skin cancer cell, and here's the first cell, and the cell then divides into two, and those cells divide into two, and you can imagine this is cut off, but these cells will all divide out in a branching fashion, and as they divide, if you're a cancer cell, you change because more DNA damage events happen in the DNA. The DNA alphabet gets altered proteins are produced, and cancer cells then become more and more aggressive with time. So that's why you hear that one patient has an early stage benign tumor, another patient might have a later stage very aggressive tumor. It's because more time has happened for these. This is called clonal evolution. The evolution, you could put a single cell in a test tube and you can follow this happening in a test tube. But if you apply treatment, so imagine these cells are just dividing normally and undergoing normal changes to their DNA. Sorry, not normal changes because it's a cancer cell, but progressive changes to the cancer cell and becoming more varied with time. So if you do a slice through any one time, you've got more variation happening. But then you treat the patient, and of course, so here's let's say here, this particular drug, and it's killing all the cells apart from the red cell. So the red cells will then come through and repopulate the tumor and perhaps metastasize. And here's more changes coming through. After several treatments, this cancer might be the recurrent one because it's coming back after all the other cells have been killed. So what we need is we need to be able to treat whole populations of cells. It's complexity in a single. So this is a single cancer behaving in this way. So this is very like, the biologists will probably see, even some of the chemists or others will recognize this picture and this man. So this is, of course, Charles Darwin, who originated the laws of evolution of species, and this is his famous diagram, which I've got on my study wall, actually, not the original, sadly, but I've got a copy of it, and this is where he said, I think. So he thought that this is how, starting from here, how a species evolves and branches, and you've got apes and so on coming out at different points in the tree of life, as it tends to be referred to. So if you turn that around and apply it, this is exactly the evolution of a cancer inside an individual patient. And the rules and the biology rules, it's survival of the fittest, overcoming the challenge of treatment or the adverse environment and becoming more aggressive. And so that's the challenge that we face. And to make it look even more remarkable, so what we can do now is follow cancers by deep sequencing. So without going into the details of DNA, it's the 4-letter code. It's what encodes makes the alphabet of life. It encodes the proteins, and if you have mutations and damage in the DNA, you produce abnormal proteins. You can follow the progression of a cancer by sequencing the DNA actually at the single cell level. So it took years and years and billions of dollars to sequence the human genome, but now we can sequence the entire genome of a cancer cell overnight and have a readout of exactly what the precise molecular damage is to that cancer, and therefore know, hopefully, how to treat it. And if you just look at this, look at the tree of life there, and then look at this. And when I tell you that this is the DNA sequence with time, or the DNA pattern with time, of two different patients, they start out with a single point here, and then you can see their evolution, and that evolution is followed by DNA sequencing of the alphabet code of the cancer. And we can now do that, not routinely, but certainly in research trials, and follow how, and this, for example, if we say here, this is a mutation that might lead to resistance. So this particular branch of the tumor no longer responds to therapy. And ideally what you want to do is to treat what's called the truncal mutations, because if you kill the whole thing early on, then you'll be likely to be more effective. So that's the challenge that we face. That's the biology that we face in this disease. How do we treat it? Well, initially we treated it with poisons. There's that awful expression which is poison, burn, and whatever the thing is for surgery, cut. One of the first treatments, and some of the first treatments to come out of my institute were based on mustard gas. So, the chemists will recognize that this is sulfur mustard. This is the dichloroethyl. You've got sulfur here in 2 dichloroethyl groups, and these are chemically reactive. That's why it killed the poor soldiers that were exposed to this in the First World War. But it was observed, it was observed that sadly the soldiers who died from this had very low white cell counts, so they looked like their bone marrow had been attacked by the poisonous gas. And it was recognized that therefore that could be used to treat leukemias. Very sort of low-level extrapolation, not very sophisticated, but it kills normal white cells, so maybe it will kill some cancer cells. And that was tried out by these two brave pharmacologists. And this is a little bit of chemistry. This paper was published in Science, which is one of the top journals in our field, in 1946, before I was born, and only just actually. But what this shows is this is a nitrogen mustard rather than a sulfur mustard for the chemists who are here, dichloroethyl amino group here. And basically these two arms can both react in that nucleophile, electrophile fashion to produce two reactive groups, and actually it can cross-link the two arms of DNA. It's not shown very well here because they hadn't figured it out completely. But we now know that these poisons kill cancer cells by cross-linking the DNA, by one arm reacting with one side of the duplex and the other arm reacting with the other side. And actually at the Chester Beatty Labs in the Fulham Road, we developed discovered and then developed 3 drugs based on this, much safer forms, less chemically reactive, more drug-like, melphalan, busulfan and chlorambucil, that are still used today actually for cancer treatment. So some of the first cancer drugs killed cancer cells by reacting chemically with the DNA. Of course, as you can imagine, that in turn caused further mutations and often led to second cancers, and also led to rapid evolution of resistance. But nevertheless, this is an effective early form of the first form of cancer. This is the type of cancer drug treatment called chemotherapy, because it's the chemo treatment that tends to lead to hair loss and problems with the bone marrow and infection and so on. But it was the early form of cancer treatment, and it is still used. So the whole thing was revolutionized during my lifetime. So I was born in 1952, and the Watson-Crick double helix of DNA was published in Nature in 1953, a year later. And then come around 2000, we are on the front of 2 of the most famous journals in the world with the human genome sequence. And the human genome sequence was, as the next slide will show, was imagined at the time. These are 2 of the great pioneers. Time magazine said that, because of the sequencing of the human genome, medicines will become pouring off the conveyor belt of research labs and into companies, and this will be the cure for cancer, etc. It probably hasn't delivered as much as we hoped. It's been hugely powerful. Knowing the genome sequence helps us to identify rare diseases, to identify different types of cancers, to understand it. Cancer is the area where the treatment of human disease has most benefited from a precise knowledge of the genome sequence. Because if you know the sequence, that's the DNA sequence of all 23 pairs of your chromosomes, and then you do the sequence of the cancer in the same patient and compare the 2, you know exactly what the differences are. And you can then, ideally, at least in 5 or 10% of cases, you then can have a drug that's been designed specifically to work on that particular patient. This is personalized medicine, which I'll come on to. So here it is. Here's a slide that I modified from one that colleagues at the MD Anderson in Houston developed. So imagine that in the days of chemotherapy, I guess times were grey then, and so here you've got a grey population, are all the same, all the patients are the same, they just have cancer, and they all get cytotoxic chemotherapy, and some respond and do quite well. Childhood leukemia was almost cured by cytotoxic drugs. Testicular cancer, 95% cured by cytotoxic therapy. Breast cancer doing very well, but most of the other cancers not so well. GI cancer quite well. But you're not doing anything about exploiting. By the way, this funny circle here, this is a way that you depict the human. If you ever see this anywhere, this is a representation. It's the way scientists show a human genome sequence, and these changes, these little things here, marks, are where there's an alteration. So this is a patient that got lots of changes to the alphabet of their DNA. So you do either a whole genome sequence or maybe more limited testing. You can have little smaller tests that look for a smaller number of genes. That's also becoming quite common and is linked to routine care actually now. But through that you can identify different types of patients. And the different DNA sequences that you find can either be predictors of response, so you might have a marker that says how well a patient will do even if you don't have any treatment. You could have another marker, another gene, a signature perhaps, that predicts whether they'll become sensitive or resistant to a particular drug, and also markers that will predict the side effects. There's another one we can add now, which is how the patient will become resistant to the treatment. So of course the idea is that you have a pill for every color. So you know the pink patient gets the pink pill because they have a particular gene X, and the blue patient gets another gene mutation, they get drug Y, and so on. So the idea being the right drug for the right patient at the right time. I think we can only offer this currently for about 5 to 10% of patients on the NHS. Not that we can't do tests, but if you could do a test, we only have drugs for about 5 or 10% of the particular types of damage and gene mutation. But here's an example of this, and I'm proud to say this was a drug that I was involved in the discovery of during the time when I led the biology of this project at AstraZeneca between 1993 and 1997. And this is for the chemists who are here. Oops, done it again. Chemists who are here. This is a quinazoline compound that's an inhibitor of a kinase. A kinase is a switch in the cell. It's the way that controls cell division. And what happens in a subset of non-small cell lung cancer is that this is the genome of a patient with a non-small cell lung cancer. They have copy number changes. They have point mutations. That's a little change in the alphabet. They have big changes like chromosomal, whole rearrangements of chromosomes getting mixed up and joining with each other. And so you can sequence that, but within there you can find a particular change which is called the epidermal growth factor receptor. And if that's mutated, the switch mechanism is forced on. The accelerator of the car of the cancer is fully on. And what you want to do, because that DNA change has led to that change in the protein machine in the cell, you want to inhibit that machine. I'll show you how we do that later on. So we came up with a drug that did that by running screens and then doing chemistry, medicinal chemistry. We produced a drug that became known as Iressa, and is now, together with another drug of that type, widely used, particularly in Japan, actually. And what you notice here is this is when the drug was. So these mutations were actually only found after Iressa was used, because the genome sequencing only came in after the drug was developed, in fact. So patients without EGFR mutations were given the drug, and you can see either those patients don't do any better than the control group. Whereas here's the survival. Sorry, I should be telling you this is a survival curve of survival over 5 years, and the proportion, this is 100%. So you can see the patients that have got the EGFR DNA damage alphabet change and are given the EGFR inhibitor drug are having that gain in at least a few years in survival, and that can be combined with other treatments. So this was together with a drug called Gleevec that was used for a particular type of leukemia that was discovered by Novartis at about the same time that we discovered Iressa, were the first targeted drugs together with the antibody Herceptin for breast cancer. These are the first examples after the genome sequence of targeted therapy. You only give the patient the targeted drug where there is a DNA change that indicates that that's the patient that will respond, and the other patients have to be offered something else if that's available. So that's one of the first targeted therapies. There's another type of change that goes on in the cancer. If you imagine, very simply, a cancer cell is like a car that goes out of control. And there are two ways that the car of cancer get out of control. There are changes that of the type I've just shown you, where the accelerator is jammed on, and the signal says go, 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 or divide, divide, spread, and invade. And what you need to do is block the accelerator being switched on. There's another type of change that happens when DNA is damaged, which is you lose the brake. So here the car is out of control because you've lost a gene or a protein that normally says stop. And if you lose that, it's the same as having your accelerator on, of course, and sometimes you have both. So here's the first drug that wasn't developed by us, but we came up with a mechanism because Mike Stratton in our institute isolated the BRCA2 gene, and BRCA1 gene had been identified by Myriad, and these are the two genes that cause most of inherited ovarian and breast cancer. And so first of all, that led to better diagnostic tests for families if you had inherited, and you could make decisions about having surgery and when you have your family and so on, which is very powerful. But in our institute, researchers showed that if you take these BRCA mutant cells and expose them to a fancy sounding drug, or comically sounding drug called a PARP inhibitor, of which olaparib or Lynparza is one, if you've got a BRCA mutation and you treat the cells with a PARP inhibitor, because they've lost a particular function, which is actually DNA repair, those cells die very dramatically. This is, by the way, familial testing in a family. And this is, so what happened was that working with a pharmaceutical company called QDOS, which was then acquired by AstraZeneca, we did the first trials of the PARP inhibitor olaparib, that's this one, in patients, having shown that cells with these BRCA mutations respond to the PARP inhibitors. We then did trials in patients with exactly that same abnormality, and they responded very well to the treatment. And olaparib was approved for ovarian cancer, and 3 other PARP inhibitors are now approved. So this is a great story, I think, of discovery. First of all, understanding a gene. This is a break gene, the BRCA genes. Then doing genetic testing that's very valuable in its own right. Then coming up with a drug treatment that kills those particular cells that have that particular abnormality, and then working with our hospital colleagues to get the drug through into clinical trials, and then working with colleagues around the world to get them approved. So these cells die by explosive DNA damage, because the BRCA gene is a gene that controls the DNA, and if you lose the BRCA gene and then you give a PARP inhibitor, you get this type of very dramatic response. This is also from, this is Paramiz Thavazu in our institute, showing a picture of an exploding triple negative breast cancer cell, probably with a BRCA mutation, responding to treatment, very dramatically illustrating how cancer cells can be killed by these types of treatment. So what about more recently? Well, over the since 2005, which is a time when we often go back to, because it's a time when the things really started to motor in our institute and also elsewhere. I can't claim that we're the only people doing this at all, but we built up an academic drug discovery group, rather unusual actually, not a biotech or a big pharma company. And the idea was that there were so many cancer genes coming out, and industry couldn't cope with even a fraction of these, and would tend to go for the ones that were more likely to be the more successful. And so we decided to go for the risky, very early approaches, the idea being that if we found new ideas that worked in cancer models, making tumors shrink in mice, say, and then early trials in humans, we then license them onto biotechs and industry, and in that way contribute to what's called the valley of death, that early stage when there's a gap in the funding available. So what we've done, that's a 2010 one that we often talk about, 20 drugs discovered over that period. 10 of these have already gone into phase 1 clinical trial, and our drug abiraterone for prostate cancer has been licensed, and hundreds of thousands of men worldwide have been treated. It's actually a multi-billion-dollar drug as well if you use American pricing. And partnership, as I'll show you on the next slide, was absolutely crucial. Through this success, we were very gratified and humbled, rather, to get the Queen's Anniversary Prize for our drug discovery research just last year, and collected it from the Palace. I think there's a slide on that coming later. But here's abiraterone. Now, this is a very busy slide. I apologize for this. It's one I use with scientific colleagues, obviously. But prostate cancer is driven by the male hormone testosterone in most cases. And abiraterone is a drug that blocks the production of the male hormone. So it blocks the production of testosterone in the adrenal gland and in the prostate cancer tissue itself. So just as in women, breast cancers are driven by the female hormone, estrogen, so estrogen receptor positive breast cancer, and then you treat with drugs like tamoxifen and anastrozole, which block the estrogen receptor. In men, large majority of men who have prostate cancer, that's driven by the male hormone. So we developed an inhibitor. It's an inhibitor of, for those who might be more expert, it's CYP17 inhibitor. It blocks one of the P450s that are involved in making hormones. And when we gave this to men with prostate cancer, against much disbelief in the community who thought that at this stage of the late stage cancer was no longer driven by the male hormone, in fact. So the biology wasn't believed. And it was also thought if you make a drug that inhibits P450, it'll be way too toxic. It'll block all the hormones around the body and you'll have a very toxic drug. And we were able to show before anyone else, before industry got involved in this, that the hypothesis was correct indeed, that men with prostate cancer, their late stage prostate cancer was still dependent on the male hormone testosterone, and then went on to show that if you trial one of these agents and give support hormones, you don't have the toxicity. It's a well-tolerated drug, and we went on to show dramatic improvement in survival. This is some of the early survival figures showing 50% increase in survival. We have men with who come to see us often with been several years on abiraterone, still responding to treatment. And you can see it went through approval in the various US and Europe by NICE a year or so later, and is now being used much earlier in the disease, actually sparing men being given a cytotoxic therapy. So if you have now, you don't have to go through having a horrible chemotherapy drug. You can get abiraterone right at the beginning. So how do we design drugs? Well, you might like to see this. So this is me, I have to say, for the Horizon program, pretending to design a drug in this. So what we're seeing is, so when you make a drug, it's like a lock and key. So imagine this is a cancer protein. It is, in fact, one cancer protein. Oh, sorry about this. I have to go back and make it move for you because it's really cool when it moves. There we are. So imagine this is one of the accelerated genes. So you've got a damage to the DNA, remember, a mutation that causes a change to one of the accelerator proteins. This is one of those accelerator type proteins. And that protein, which is what causes the cancer after the DNA damage, is a globular shape. You need to design, if you want to block it, you need to get a drug that fits into this little bit, which is where the protein does its damage. And here you can see me trying to design and point out in our imaging suite, almost like a lock in a key. You're trying to fit, this is the cavity in the protein is the lock, and the protein is the key. And you can design that with exquisite accuracy. Just moving around for you so you can see the different way it binds into the active site, and you'll shake around a little bit and see some of the ways that the protein forms hydrogen bond interactions, if any chemists are here, with the target protein. There's the network of hydrogen bonding interactions. So it fits into the shape, and then it makes these bonding interactions, and it blocks the function of the protein. So if you like it, someone wrecks the lock, it puts super glue in the lock of this particular target protein. And that's the way we design drugs these days. We often do a big screen, first of all, to find precursor drugs, and then using the crystal, I should have said, this is the X-ray crystal structure. So you use powerful X-rays to get the 3D shape of the target protein, project them onto a screen, do computer graphics, and find molecules in the computer that can block the function of the protein. Here's an example of one of these that we've just to show you a couple of examples. This is a local lady close to Sutton who had ovarian cancer. Ovarian cancer is a very nasty disease. It's often called the silent killer. It's not detected very well. Patients present quite late, and they're given chemotherapy, which is rather toxic, and many relapse. This is a drug that we designed to be taken up by the particularly by the ovarian cancer cells through a particular mechanism. It's called the alpha folate receptor for the enthusiasts. And this drug has been taken, so nobody believed in this idea either. We developed it on our own. We took it into clinical trial in the Marsden. We saw a 70% response rate in phase 1 clinical trial of ovarian cancer. These are women who are no longer responding to the chemo that's available and would have no other hope, and are seeing a 70% response rate. We licensed this recently. Having shown that it works, we've licensed it to a small company, island-based company. Perhaps there'll be more of those with recent developments, I don't know, but better keep off that. But this company is now going to take these into large trials for clinical trial. I should have said that when we developed abiraterone, look at the number of organisations that are involved here. We and Cancer Research UK started out doing the project. We licensed it to BTG, a sort of venture capital outfit at the time. They then gave it to Cougar Technology, a small biotech company in the west coast of America, and then eventually the drug was licensed to Janssen, and it's now a blockbuster for license. But it started out in the lab of the ICR and went through those different partners. This is the partnership idea. And here we've gone through several different partners over time and are now working with with Carrick to take this through to hopefully approval and widespread use on the NHS. So that's a nice picture. I'll show it really not because it's to brag particularly, although it is nice, but this is my successor, Raj Chopra. We went up together to get this, and Raj is now directing. Since I took over as CEO, I gave up running the drug discovery, and Raj is now running with his own ideas and own exciting new ideas. And what was very nice is that the Palace asked us to take a selection of PhD students and postdocs and young scientists, as well as one or two of the older guys like me and ladies, and there we are celebrating across the junior seniority divide outside the Palace on a very chilly, I think it was February day last year. These are just snippets of 3 more drugs that we're taking into clinical trial. Again, this is a little bit for if there are any aficionados here. So we've got a drug called an MPS1 inhibitor. It's in phase 1 clinical trial with a company called Boston Pharmaceuticals. Again, it's a young little biotech that's taking our drug forward. These patients are being treated not in Boston, but in Sutton and around London, actually. Our CHEK1 inhibitor, also with a Californian biotech company, also in clinical trial with us in London. And our PKB inhibitor, which we developed with a great company called Aztecs in Cambridge, we've licensed it to AstraZeneca. Well, actually, they finished the design of the drug with us and after us, and then that's in showing very interesting activity in triple-negative breast cancer. I'll show this one because it may be the last project that I'll be involved with in my career. I don't know. It's still got a way to go, but this is an idea that my lab had for blocking this particular accelerator pathway. It's more of a support pathway, actually, that cancers need to continue to divide. And nobody believed in this target, and we worked a little bit with AstraZeneca. We now have a drug, as you can see, that's going into clinical trial this summer for ovarian cancer and also myeloma. And here's just a prototype of the drug. We're growing human ovarian cancer cells in mice, in special mice that will tolerate a human ovarian cancer growing there. And this is the group that's not treated. And here you can see the much slower growth in the group that's treated. And we do a minimal amount of that sort of work before going into clinical trial. So we're excited about that. Hugely exciting. I'm sure you can't have failed to have seen the enormous publicity, and rightly so, about the science of immunology. Cancer cells are because they have these DNA changes, they have different proteins on them, and so they can be recognized not quite like a virus or a bacterium, but certainly different from the normal cells of the body. And if the immune response in the patient is functional, you'll get things like, in particular, fashionable at the moment, or very effective at treatments to activate T cells. These are T lymphocytes that recognize the cancer and can kill the cancer. The trouble is, through that Darwinian evolution mechanism, cancers are very clever, if you want to put it in that sort of teleological way, because they develop a method that blocks the activated T cell from recognizing the target cell. So the new treatments, and this is the ones that are highlighted here once again on Time magazine, the new immunotherapies that led to dramatic responses in melanoma. We're now seeing activity in patients with lung cancer and many other types of cancer as well, is it blocks the blocking mechanism. So the cancer has disabled the activation mechanism and blocked it, and then an antibody has been designed that overcomes the block, releases the negative handshake if in a way, and gets the lymphocytes activated against the tumor cell. That's just one snippet of how many of these approaches, these CAR T cells, for example, are engineered cells to make them more effective. And this is, you probably heard the story of the miraculous responses of young children with leukemia to these treatments. But all of these different types of immune responses can be activated against cancer. Viral therapy, cellular therapy, different lymphocytes, NK cells, and so on. And here's another of those great pictures from our competition. This is from Matthias Crespo in Johan de Bono's lab. And this is a type of liver cancer, actually a bile duct cancer, where the tumor cells are in yellow, and the green and red cells are the lymphocytes coming into the tumor, recognizing the cancer and killing it. And when you give one of these immune activating responses, you see many more of these lymphocytes coming in. But some patients don't respond in this way. So some patients get lots of their immune response being activated, and they have a dramatic response and cure, actually sometimes from a very small number of treatments. Real cures, long-term effects. But some patients don't respond at all. They have what are called immune cold tumors rather than immune hot tumors. We're trying to understand what makes the difference between a hot and a cold immune tumor and design better drugs that will take advantage of that. But this is a tremendously exciting area. We're making new drugs in this area as well. I want to tell you, just as we close, I just want to tell you a little bit about a truly remarkable type of radiotherapy that we're now giving, which is a machine called the MR-Linac. I don't know whether there are any physics people here, but this is a truly amazing machine which is being developed by Elector, which is a company in Croydon that makes linear accelerators, working with Philips, which makes MR imaging machines. What you're doing here, you'll be familiar of an MRI scan, which is a high magnetic field, usually 1.5 Tesla magnet, which produces an MR image, which locates where the tumor is. And often, prior to this new technology, that machine would be used to find where the tumor is, and then maybe a few weeks later, the patient would come in and be back on the table, and hopefully it's better than this actually, but you hope that the pattern is still the same, and you radiate what was seen maybe a week before or a few days before. Moreover, it doesn't account for movement of the patient. Patients have to breathe if they're having their lung tumor irradiated. So what this does is it joins together in a single huge machine, actually the size of this room, a high field MR machine, a magnetic resonance imaging machine, that's joined physically to a linear accelerator, which is the machine that produces the X-rays when you have a radiation treatment. And interface between the two is a highly sophisticated computer, because believe it or not, in a high field MR machine, if you go near that with metal, if you've got change in your pocket, you'll fly across the room and be dragged thereby. So you have to have a setup where you can have lots of this metal next to a huge magnet with a high magnetic field. Moreover, a high magnetic field bends the radiation beam, believe it. I couldn't believe this when I was told that it's such a high magnetic force, the field, that you have to correct for that as well. But the idea is that you have in real time an image taken of the tumor, and it's being irradiated at the same time, not a week after or a few days after. Moreover, it takes into account the movement. So let's just see how this works. So this is the first patient actually in the UK in our hospital. First patient in the UK was treated in our hospital. A gentleman with prostate cancer. This is from the BBC film that was made about. So here's the patient going into the, I hope he's not claustrophobic like I am, because I would not enjoy that. But there are machines that will be developed with, dealing with that. So here's the patient, and here's the technological and the team of doctors looking at the, and what they're doing, that's the tumor. Sorry. Well, we've gone back to the beginning, forgive me. But if you just tolerate this for a second, you'll see that in real time they're able to get an image, an MRI of the prostate cancer, and then they're going to draw, almost like a kid playing on a computer and drawing a Photoshop around the tumor. So there we are. That's the same patients. There's the prostate cancer that you'll see in a second. There it is. And what they'll see is that it's gone negative because they're moving off a little bit that they don't want to irradiate. You don't want to irradiate the nerves and the normal tissue around the tumor. You want to get all the dose into the tumor. And that's a simulation of how the dose will be delivered, and also showing the movement actually through as it goes through the tumor. So he's been irradiated and he's leaving the... but I can show you in a model, which I find totally remarkable. So now we irradiate. This is an actual model of a tumor being irradiated. The two tubes that are going up and down are where your airway separates into the two lungs. That's a big tumor in the lung. It's obviously speeded up because you don't breathe as quickly as that, but as you breathe, that tumor is moving up and down in the field, and the orange line is the radiation beam following the movement of the tumor. I think if I was going to have radiation of my prostate or anywhere else, I'd like it to be tracked by a machine that followed the movement and delivered precisely to the cancer. So these machines are being rolled out. I think they will in turn be available on the NHS. It's just in clinical trials at the moment. This is fantastic technology and has the potential to revolutionize the treatment of cancer with radiation. And here's another of those wonderful pictures. This is now looking at cell death after cells being killed by radiation. So here's some cells that are stained, as you like. So these are normal cells, and these are cells that are, you can see the wreckage here, these are irradiated tumor cells. Not only that, not only are these highly damaged cells that are dying actually, but once again the yellow and green cells are the immune cells coming in. So what actually happens when you irradiate a cancer is it makes the immune response even more effective. So the immune response combines with the radiation to kill cancer cells more effectively, which is great. There's an incredible machine that's by my colleague Aradazi at Imperial College, which has the potential to revolutionize surgery. And this is what's called an intelligent knife, or a sniffing scalpel, sometimes called, because it's, as you can see, the scalpel here, where the patient's being opened up, is linked to a tube, and this is linked to what the chemists call a mass spectrometer. So some smoke is generated when the heated knife cuts into the patient. The smoke goes up the tube onto the mass spec and analyzes what's in there and can tell the difference between the tumor and the normal cell. So when the surgeon isn't quite sure whether he's still in the cancer or moving into the normal tissue, you can be guided by this so-called intelligent knife. Still in early times, but even surgery now is going to be revolutionized. I just want to finish with a couple of comments about access and cost. And questions might come up about this. So we did if you're interested in this policy side of things, we did a review which we've just published on our website. Just put patent to patent and you'll find it freely available on our website. What we show by analyzing drugs that have become available over the last 10 years or so is that there are big delays in clinical trials and the approval of drugs. So patients on the NHS are not getting drugs fast enough, in our view. A lot of it is bureaucracy, and we think we can help with that. Moreover, there's huge variability in the availability of drugs against tumor types. Hardly any drugs for children. In fact, no drugs approved for children for a decade. No drugs approved for brain cancer for a decade. So huge areas where drugs are just not coming through. So that tells us what we need to be doing in the lab. And moreover, although NICE is doing a great job of getting more drugs through, it's taking longer, and more innovative drugs are actually not being prioritized. So it's taking longer to get drugs through, and the more innovative drugs, for some reason, the riskier but potentially high payoff drugs are not being prioritized. So we think that's a real concern, and we've got ideas about how to work with industry and colleagues to develop that. And then there's the other thing. So here is a patient that, it's a little bit, it's a painful slide to show in a way, but humorous in another way. So the patient's linked up to a calculator that's telling them how expensive the treatment is. $150,000 typically for a immune therapy. You combine 2 or 3 drugs together, you're into the cost of a pretty large house. In fact, the cost of cancer treatment in the US now exceeds the cost of an average house and is the cause of bankruptcy in many patients. And thank goodness we have the NHS, but we need to do even the cost of drugs for the NHS, of course, is troublesome, especially in combinations. Again, we've got ideas for how we might work together with industry and policymakers to maybe do something about that. Early detection and diagnosis. We and others are developing gene tests. So I quite like this. The best thing you can do for preventing cancer is to exercise, to not be overweight, and I'm afraid to decrease your alcohol. So lots of the stuff you would prefer not to do, you have to do for prevention. The doctor here is saying, what fits your busy schedule better, exercising for an hour a day or being dead 24 hours a day, which I find it's quite a good way of looking at this, and I need to do it myself. So better test, but I think the way forward, screening for everybody. PSA is not great for prostate cancer. Mammography helps with breast cancer, but is not 100% accurate. we're trying to develop gene tests that will identify individuals that are higher likelihood of risk, and they're the ones that you can do more intense monitoring on. In fact, we are developing, Roseals in our institute is developing a saliva test, a spit test as it's called, for high-risk prostate cancer. You spit in a tube, gene sequencing is done in the saliva, and then a group of genes have been identified that will tell the doctor if the pilot experiments work. This is a high-risk patient that should be monitored much more often. Low-risk patients can often be left with their very early-stage prostate cancer and have no side effects. With breast cancer now, we run the biggest study looking at genetic, lifestyle, environmental factors, looking at, it's actually 130,000 women for 40 years and following these gene changes and their hormones and so on, and looking for what leads to better ways of identifying the women who are more likely to get cancer. And we can imagine in the future, and we're working with Imperial College on this, better detection tests that will be, a wearable device that will speak to your iPhone and tell you that it's time to go and have a test with your oncologist. It's super, it's science fiction, isn't it? But in some ways it's awfully close from some of the examples that I've shown you. I just want to show you a couple of things that we're doing at our institute to finish. First of all, we need to develop more cancer drugs, so this is our new 70 million cancer drug development building. It looked like that today. It's got the roof on, the windows are in. It's a fantastic new facility where we hope we can develop more of these evolution-busting drugs. I should have said that one of the ways to block that branching evolution is to give combinations of drugs so as to take out several different branches of the tree. So we're working very hard on that aspect as well. Working with our cancer evolution team and our drug discovery team to tackle that particular problem. And then the other thing we're doing, just so you can show how we link up to industry, is that with the local council, we've formed a partnership with the Sutton Council where we're going to put a biotech park on the land we've acquired from the NHS from the old Sutton Hospital. And you can read the details here if you're interested in the more commercial side. Tons of space, state-of-the-art facilities, spin-out companies, medtech, biotech, so we can do more of the type of work that we want to do and get things through quicker, generating value to the economy, and we think it'll be a great environment for attracting top talent into London. It shouldn't just be Oxford and Cambridge that are doing the biotech companies. We should be doing more companies in London as well, because there's great talent here. So that's an exciting thing for us to be doing. So I'll stop there. I hope I've, and sorry if I've gone on a wee bit long, I hope I've given you a sense, first of all, that we do understand how cancer is caused. It's disease of genes, and we can identify those genes. We've got good ideas of how to combat at least some of those genes, and that's led to great improvements in survival. We can do better radiotherapy and target the tumor more effectively. We can even do surgery in a more precise way. So I think the future is bright. We shouldn't underestimate the scale of the challenge because of that ability to adapt and evolve. But we have ideas how to combat that. So I hope that's given you, I think, a realistic but hopeful view of cancer. And thank you for coming. Welcome your comments. Happy to take any questions now or over drinks afterwards as well. Please, yeah. The one at the back next.
Thank you for your talk. It's really fascinating. Can you hear me?
Yes. Can everyone else?
Sorry, this isn't working.
Is the mic on?
Raise my voice. Can you hear me now? No, it's not. No, this saliva test.
Yes. I'll repeat it for you. I'll repeat it. So go ahead.
Is this saliva test happening here and now?
It's being tested in GP surgeries now, yes.
And at what age do you do that?
I think it's being obviously tested in older men because the frequency is much more common in older men. So it's the age at which, I think it's 40 plus. I don't know the precise age for that particular trial, but we have experts who do know that. But I should say it's still in the early trial stage, but we've identified the genes already through sequencing the DNA of individuals taken from their blood. And what we're now doing is, and we've identified which genes, when wrong, most predisposed to the most aggressive type of prostate cancer, the really nasty cancers. you'd probably all be familiar that many men that have a PSA test, they go along, they have a rather unpleasant procedure, and actually they don't have cancer. And similarly, some men that do have, so many men that I would reassure you, many men do have your PSA test done because it does pick up cancers, but many men that have that don't have prostate cancer. So this would be a way of... Correct, it's a false positive, yeah? But also there are false negatives, so there are some men that have prostate cancer that don't have the high PSA. So what this will do is it will, and the test is expensive and it's not very effective. So if we can identify that, and also this is very interesting, that many men that have more benign prostate cancer actually don't need to be treated because their cancer will never grow to be troublesome. And why have the risk of surgery that might lead to impotence and loss of, you know, urine control and so on, when your cancer won't do any harm. So what you want to do is to identify those prostate cancers that are the most likely to be the most nastiest. And so we found those genes. We can now pick up those, believe it or not, you can sequence the DNA in saliva because there are cells in the saliva, and so you're doing a DNA test. And now what we want to see is does it work in a GP surgery? So 5 years probably from now, that might be, if it works, might be a routine test.
Thank you very much.
The gentleman at the back, and then there's a lady over there.
You mentioned that you could grow ovarian cells in mice,
Yeah.
but then went on to say we try not to do very much of this work. Does the criticism of the Animal Protection League hamper your work?
It doesn't.
How
No,
are you trying to mitigate that?
Yeah, thank you for asking that, because and I always make a point of saying that we do animal research, and it is essential, because there are some things where you can model cancer cells on a computer, and we do that. There are others where you can model cancer cells. We had a big publicity over a paper we published in Science, one of the top journals, where you can grow mini tumors in a dish. You know, you can grow, we call them organoids, small organs of the tumor, and we can treat them. But there's, you can't fully capture. You saw the complexity of cancers in those wonderful and terrifying images. So you have to do the animal experiments. I didn't actually say, what I think I said was we minimize the amount of that testing. I always make a point of saying we do animal testing because it's essential in cancer, but we minimize the amount that we do so that we know enough to progress it to patients. But no, I mean, these days, you know, everyone has a right to their view, of course, but the impact on cancer research is, I think, not where those who would be motivated to you know, go against would not choose cancer tend to be as its target. So we haven't, and I think I would like to say, and I've been the author for now, I shouldn't, this gives my age away as well, I think it's now 3 lots of every 10 years I've done, published the worldwide used guidelines for using animals in cancer research, and it's the 3 R's, you know, reduce, refine and replace. And so I think judicious use of animal models for cancer is absolutely essential. I would get that point across. But we obviously would minimize the amount that we do, and why would you not minimize it? It's the obvious thing to do. And we do most of it in mice, most of the work that we do. But thank you for asking that question, because I think it's important that it's discussed. And we're very open about the work that we do. And I think that's one of the reasons why I think society perhaps has become more accepting of it, because they see the value and the need. But also, if we're open about what we do and we show images of the animals that we do, we invite people in to film and so on. So, you know, I think that's a much healthier way. Thank you for the question. I think there's a lady on the back row on the right.
Thanks. So you mentioned some of the financial sort of difficulties or challenges that you face, and I was just wondering, how do you balance perhaps some sort of tumours and types of cancers that have massive sort of charity and like maybe media coverage that are really well known, such as like breast cancer? How do you manage finding cures for the ones that perhaps affect less people but aren't as like currently our methods aren't as effective?
Yeah, great question.
What are the challenges?
Great question, and we are doing that. So there are a number of ways that we do that. So the first thing to say is that there's still a lot of breast cancer that doesn't get cured. There's still a lot of prostate cancer. There is a still need to work on that. So we get a lot of funding from Cancer Research UK, and they have for some time been leading the way, I think, and we have as well, in saying we need to cover more cancers than we do. The Tessa Jowell experience, I was in the room actually when Tessa gave her first, not the last speech in Parliament, but when she gave her last few words to a group of scientists and politicians in Whitehall, and that's I think that has beginning to lead to a movement, just as we had with breast cancer advocates and prostate cancer advocates. So there is still funding and still work to be done from Prostate Cancer UK, fund our prostate work, Breast Cancer Now, fund our breast cancer work, and there's still some pretty horrible breast cancers to be treated. But for example, there's very little funding going into children's cancers, and what we work very closely with the children's cancer charities who fund much of our work, for example, into, I have to say, I find the notion of cancer is a pretty nasty disease, but the notion that it develops in children and even in babies, to me, is a cruel and horrible thing, and I've certainly devoted quite a lot of my own time and efforts to work hard on that. There have been no new drugs approved for children's cancer, and not enough work done on it. So we are now working with the children's charities and also with the Brain Tumour Charity, which runs brain tumour research, and we're working on children's brain tumour in collaboration with colleagues in Cambridge. That's one example. A lot of that, we've just founded a sarcoma centre. Sarcomas are tumours of muscle, and they can be very, or commonly of muscle. And they come in many different subtypes, so they're quite rare. There's a type called osteosarcoma. There's probably a few hundred cases a year of that. But we've, with philanthropic support, we've created a sarcoma centre, and we now created a small group that's working on sarcoma with a particular group of a family that had a loss of a young, relatively young woman from that disease. And we raise the budget for my institute is about 130 million a year. It's a lot, but it's not enough. And of that, we raise about 16 million from philanthropic giving, and we tend to put that funding, which is not linked directly to a charity, we tend to put that into the more difficult, we channel quite a bit of that funding. So that's the way we, but it's a complex thing. In some ways, scientists will follow where the funding is, because how can you work if you're not funded? But institutes like ours, I think, have to create funding mechanisms that allow those cancers to be covered as well. And the pendulum is swinging. So there's much more. This year, there's a huge amount of research that Cancer Research UK and the Brain Tumour Trust are putting into brain cancer, which is a terrible disease, with a terrible survival. So, sorry, a long answer, but does that sort of more or less describe it? Yeah. Don't underestimate the power of families, survivors, advocates, in raising awareness. The HIV community led the way, the breast cancer community and prostate cancer community as well. Now we're seeing the brain cancer community, some fantastically active advocates for children's cancer, and they're fantastic to work with. I think I saw, yeah.
Yeah, can I ask about the regulatory system? Everyone who's not used to it, never done it before, knows just how, discovers just how Byzantine it is, labyrinthine. Do you think they are hung up on old-fashioned clinical trial processes which aren't appropriate for cancer, where there is very rarely a placebo effect, I suspect?
Yes. So I think what you have to balance here to try and give it'd be easy for me to say red tape, holding us up, should be getting more of these drugs into patients, and all of that is true. But at the same time, drugs have got to be, you can only take them forward if they're reasonably safe. Now, the argument is that, for some cancer patients, and all cancer patients are different. I've written, I don't know whether those are interested in, those who like to talk about a war against cancer versus people using less military or aggressive language. But for some patients who want to as some would call it, fight to the bitter end, and they would take anything that's available, and if the toxicity or side effects are either unknown or less severe, they would want that. And of course there are others who would have different views. So there's a minimal amount of, or a certain amount of, it sounds like you know the area, that has to be done to check for safety and so on. But we've pushed very hard for nice, at the moment, we won't speak about what the future might hold, but at the moment, a drug has to be approved by the European Medicines Agency, and then if it's approved by the European Medicines Agency, which is taking too long, and would then come to NICE, and that's taking a while, although I don't want to criticize NICE because they've got a tough job, but that's taking too long and not being innovative as well. And what there seem to be is big gaps between the different stages where nothing seems to be happening. So I think there is bureaucracy there. It could be speeded up. And I think drugs could be approved much earlier. In the US, they have much greater use of this orphan drug status. Also they have, and NICE have got this, but I think it's only for 2 drugs a year, a sort of pioneer category, where if something really looks exciting and different, you can fast track it into the clinic. I think, and much of this is to do with cost. So here's a thing I'll comment about the cost. So the cost is done on the basis that the drug will be used in the way that it's currently given. So right now, if you're treating late-stage cancer patients, you get, say, 6 months gain in survival. If it's being sold at this cost, then the QALYs, as they call it, this is the cost of quality-adjusted life, then it doesn't pass the test. What's neglected in this is that the first approval of a cancer drug, the first getting it into patients, is just the first step, when it could then be used earlier, and it could be used in combination, and it could be used in different types of cancers, where the overall value will be greater. So I think too many drugs are being stopped at the early stage because of a sort of incorrect calculation. So I think we should try and use the accelerated approval mechanism, and I think we should use a more creative approach to cost, where initially cost isn't seen as too much of a disincentive, so that at least we get the patients in and see if they have a chance of responding, and then we can deal with the overall cost when the drug is out on the NHS. Sorry, a bit of a long-winded, but I do agree with you. There's efficiencies that can be made, there's time that could be saved, and patients could get the drugs faster. And I just hope that I'll just say this, it's not a political point, but I just say that whatever changes are in front of us, the hope is that they won't lead to a worsening of that. And whatever political outcome is achieved, we need to find a way of getting drugs through quicker, whatever the politics dictate. Thanks for the question. It's a very good one. Madam.
This is a very, very simple question. But if a woman has breast cancer, does it automatically mean that her daughters are at risk?
Simple questions are always the best questions. No, it doesn't. And even if you have a BRCA if you have one of the BRCA mutations, it means that those daughters who carry the BRCA mutation will be at risk. Not guaranteed, but will be at risk. So in the families where there's a very well-known gene that is a strong causer of cancer, like the BRCA genes and 1 or 2 others, their familial testing in the family is done. But for many other breast cancers, it's not inherited, and so it's not linked. So there isn't a one-size-fits-all answer to your question. But if you're in a BRCA family, for example, then careful testing. If the individual chooses that testing, of course, it's a choice, and then monitoring, and then if that's done, the affected woman can decide what to do about the future in terms of decisions about family and surgery and so on. But it doesn't mean that if mum or grandmum has a breast tumour, then you are at high risk automatically. It's rather more complicated than that. And just while we're waiting for this gentleman to ask his question, what we're doing is in the other cancers where it's not a single powerful break or accelerator gene that's causing the cancer, but actually a smaller contribution from multiple points in the engine. We're trying to identify what are the patterns of genes, maybe 10 or 20 genes. So the prostate cancer test, for example, will be a group of genes to see if that group of genes is able to find the most at-risk men with prostate cancer. So.
Hi. My question is, my mum, about 73 years old, she was diagnosed with uterus cancer December 2016. She'd got it diagnosed in India because she was on holiday then. And then she had to come home and she had to go through the whole, how do you say, the process, which I observed because I had to go and assist her at the the hospital in central London. A lot of the treatment that she was given, like, how do you say, I can't remember, like the chemo and stuff,
yeah.
that at 73,
Yeah.
that really messed her up. Through my research, because Indian tradition, there is this philosophy which is of the conventional medicine, but also we've got, funnily enough, quite a long 5,000-year record of natural medicines as well. And the one thing I'd been learning about a lot is on the back of my mum getting the uterus cancer, because she was bleeding for about 2 years beforehand. And to me, I was like, I didn't know what that meant. All I know is that she's bleeding. And through my research and looking at all the different herbs and everything, the one thing I picked up on was with and this is the thing that actually healed her. And within December 2017, when she went back to the doctor, or the doctor sent her a letter saying you've gone from stage 3 to stage 0. The thing that I was giving her for pretty much the whole year, apart from, in addition to raw vegan diet and lots of vegetables and everything else, and fasting, the key ingredients I was giving her was cannabis oil.
Mm-hmm.
And not the CBD stuff, which you can still get at the moment, depending on how the MHRA decides on it, but the full cannabis oil. And this is something which is in hundreds of reports in Mendelian ResearchGate, and it's attested to, it's scientifically proven. My question is that when there's so much research, testified, proven research, even sort of proof of how it used to be sold in pharmacies in the 1900s in London, and now just basic CBD oil, which is assisting people, what steps need to be taken when you've got institutes around the world saying, 2 cancer institutes in the US saying that this is already working benefit?
Okay.
What's the roadblock
Yeah.
in the UK that's not allowing this to go through when you've got people saying
Yeah.
that just basic CBD oil is actually assisting? And I'm glad that my mum got free of it, but I just dismayed when I see other people who can't get access
Sure.
to the same thing.
I think I get your question, and actually thank you for the comments. There's several pieces in there, and it's complicated. I'm happy to talk to you afterwards, but I'll try and be brief. But so the first thing is that, and I'm sorry for your mum's illness, and I'm very delighted that she's hopefully doing well. This is complicated because the first thing is early diagnosis is absolutely essential. So, you know, if anyone has bleeding of that type which is unusual, you should go and get it checked out, because the earlier you have a uterine or any other tumor, wherever it is, you will do better, whatever the treatment is. That's the first thing. Second thing is the giving of chemotherapy to elderly patients is quite difficult. Some elderly patients tolerate, because we're all biologically different, tolerate high-dose chemotherapy or radiation therapy extremely well, and others get very sick, and the doctor has to make a decision about how to do that. That's a point as well. In terms of the use of cannabis, cannabis oil, it's very controversial. There are others who would disagree with you that I'm not saying I do, but there are those who would challenge that proper randomized clinical trials where you treat with one treatment versus another and ask if that particular ingredient has, it could be, for example, I don't want to upset you in any way, but it could be that your mum was maybe responding as well to the other therapy, and maybe it added value as well. I'm not saying that it didn't, it may well. And there are drugs, cancer drugs, that have come from particularly the Chinese herbal medicine that is now used for cancer treatment have identified the active principle. I think the important thing is that, and this is where you may not like what I say, but it's important, the individual anecdotes, and it includes the use of fasting and vegan diets and so on. That works well for some people, and others would not choose to do that. But what we must do, I think, if we're going to push the broader use of any type of treatment, and I have a completely open mind of where the treatment comes from. I do think that at a certain point you need to reduce it to what the active ingredient is and try and test that in a careful way and not mixtures, because the mixtures can vary. But then you do have to, at a certain point, just as if it was a new medicine that came from the science that I've talked about, you have to do what's called a randomized clinical trial that tests a group of women or men against another group that have another treatment and show statistically that it's a real value. Because another individual might have a story that didn't come so well. So I'm sorry to say what you're saying, it may well be true, but in large numbers it may not. So I think that's the answer. And there are those who would say that the trials have been done and have not been done as rigorously. But there definitely are publications that show that some cancer cells are affected by that treatment. So I'm not poo-pooing it. I'm just saying we have to be careful, and no more careful than we would be with any other treatment that I myself had. But let's talk afterwards if you want to. It's probably better to give the other members, and then I will come back to you later. I'm very happy to. Madam.
So you mentioned that the PARP inhibitors such as
Yeah. Yes.
work by the BRCA mutation.
Yes.
I know, for example, alaprib has been approved in the EU for patients. Can you hear me? So alaprib has been approved in the EU for patients regardless of BRCA status. So I wondered by which mechanism alaprib is working in these patients.
Yes. So this is interesting. So if you have a BRCA mutation, or one of the BRCA mutations, you have a very high chance of responding to a PARP inhibitor like alaprib, and that's where the science first identified that you have this DNA defect called a BRCA mutation, and the PARP inhibitor comes in and exploits that defect and gets the explosive killing of the cancer cell. And the first PARP inhibitors were approved with BRCA mutation, what's called on the label. So, you had to have a PARP test, and if you had a BRCA mutation, then you could get the olaparib on the NHS. And when the science was done, that was correct. But since then, scientists have identified that there are other genes that, if they go wrong, behave in a similar way to the PARP inhibitors, to the BRCA. So I think that's why we're beginning to see a wider range of tumours that don't necessarily have to have a BRCA mutation, but they can be BRCA-like. There's another way, which is that if you, depending on how you respond to the platinum treatment that women tend to have before they get the olaparib, that can predispose the likelihood that you will do well. So some of the PARP inhibitors are being approved, depending on how the trials were done, still with BRCA on the label, and other PARP inhibitors now being approved that don't necessarily have to have that. So that's the current status. The science is still being done. I don't know.
Just in the interest of time.
Okay.
Is that okay? Maybe
Yes,
if you
absolutely.
could
I'm
take some,
yeah.
you'll have to stay a little bit and take some.
Yes, the advice I'm being given by the boss is that we go for a drink and chat over drinks. So thank you for the great questions, by the way. That was extremely good.
Lots more questions, so we'll have a drink session afterwards. Please feel free to stay, and hopefully Paul will be able to stay
Yeah.
for a few minutes and take some questions. And I'd just like to say thank you, Paul. Maybe if you could just wait a minute, please. Just to say thank you. You've managed to cover a hugely complex area in 40 minutes and make it easily understandable. I think that's a huge challenge in itself. I think to hear about the work of the Institute and the great minds that are working on this terrible disease, I think gives us all hope, and I think that's a great thing to see. So on behalf of SCI, and I think all of you would like to say thank you. And